In Unit 3, Lecture 1, Part 1, we're going to look at an overview of microbial pathogenicity. As typical, we begin our lesson with a list of fundamental statements. Now, beginning with Unit 3, I'm going to stop reading the fundamental statements to you and just assume you're doing that as part of your study techniques. So make sure, again, that you go through the fundamental statements here. You also notice that this is a lecture unit on your lecture schedule that says to read on your own. This is primarily a lot of definitions we need to know, but in this lecture I will go through this anyway so you can hear me talk about these various definitions. So again, review your fundamental statements at the beginning here, which are most of the definitions you need to know. And notice under your detailed learning objectives, we have a lot of definitions you'll need to know for the quiz. A few that deal with the process of disease, but again, mostly a lot of definitions. So again, make sure you're going through and answering your performance objectives and learning those performance objectives for your Unit 3 exam. So let's begin with our overview of microbial pathogenesis. The first section deals with infection versus disease. Now we're going to look at some of the things we talked about in our first lecture regarding the microbiota and microbiome just to get us into this. As we've mentioned, of course, before in the course, bacteria are found in almost every environment, but relatively few bacteria cause human disease, and many, of course, are beneficial to humans. They're important in decomposition to recycle nutrients in nature. We can use them to get industrial and pharmaceutical materials, etc. Now remember, the human body has an estimated 30 trillion human cells, most of those being red blood cells, but the human body contains about 9 trillion bacteria and other microbes. So most of the time we have more microbiota or microorganisms living in on the body than we have human cells. And these microbiota establish a symbiotic relationship between the host and the microbiota. Now remember, as we learned earlier, symbiosis is when two dissimilar species or organisms live together, such as bacteria and humans. And this complex symbiotic relationship between the humans and their natural microbiota is critical to good health. And again, we talked about that when we talked about some of the benefits of bacteria in our very first lecture of the course. Now, symbiosis can either be mutualistic or commensalistic. When the symbiosis is mutualistic, that's where the relationship between two organisms is mutually beneficial. Both organisms are benefiting from the two organisms living together. If it's a commensalistic relationship, that's where one organism lives on or within another and derives benefit without necessarily harming or benefiting the host. So our microbiota have both symbiotic and commensalistic relationships with our body as symbiosis. So remember, the microbiota refer to the microorganisms that typically inhabit a region. So of course, those that inhabit the human body would be the human microbiota. Now, the millions of genes associated with the microbiota in the human body, especially in the intestinal tract, are critical to the body. They aid in digestion of many foods. They help regulate many host metabolic pathways, and they help regulate our body's immune defenses. So without this microbiota, our body couldn't function. The collective microbial genes of the microbiota in microbiology is often referred to as the human microbiome, with the microbiome referring to the genes of the microbiota. Now, there's an estimated 3 to 5 million genes from over 1,000 species that constitute the human microbiota, compared to the approximately 223,000 genes that make up the human genome. So most of the genes that are functioning, for example, in our intestinal tract are the microbial genes rather than the human genes. So some of these normal microbiota can also cause opportunistic infections when they get into the parts of the body where they don't normally live, or when the body becomes immunosuppressed. So an opportunistic infection is an infection caused by opportunistic microbes. that usually do not cause disease in a host with a healthy immune system, unless they get somewhere in the body where they shouldn't be, or unless the body becomes immunosuppressed. So some of these normal microbiota can cause opportunistic infections. They're harmful only when given the opportunity, such as getting into the parts of the body where they don't normally live. For example, fecal bacteria getting into our urinary tract, causing urinary tract infections, or when the body becomes immunosuppressed and the microbiota can then overgrow and become harmful. So when normal microbiota cause opportunistic infections, they're referred to as opportunistic pathogens. And the infections are often called endogenous infections. So again, an opportunistic infection, as we just mentioned, is an infection caused by opportunistic microbes that usually don't cause harm in a host with a healthy immune system. And these are often called endogenous infections. Endo refers to within. So this is an infectious disease arising from infectious agents already present in the body, but previously asymptomatic, when microorganisms already live in the body and aren't causing harm suddenly start causing harm. And a good example of urinary tract infections, where when the E. coli and other opportunistic pathogens that cause urinary tract infections live in the intestines are harmless. But when they get up the urethra into the bladder, they can cause urinary tract infections. Microbes that are not part of our normal microbiota that enter the body and cause disease, we call true pathogens. And these infections are often referred to as exogenous infections. So exogenous infections are infections caused when non-commensal bacteria, those that are not living in a symbiotic relationship with the body from outside the body, enter a host. So what we're going to be concentrating on in this section are the bacteria that are potentially harmful to humans, and try to understand the factors that influence their ability to cause disease. So pathogenicity and virulence are terms that refer to an organism's ability to cause disease. Pathogenicity is the ability of microbes to cause disease and inflict damage upon the host, whereas virulence is the degree of pathogenicity within a group or species of microbes. So pathogenicity, again, is simply the ability of an organism to cause a disease or inflict damage on the host. And this is often due to virulence factors that it produces. And virulence factors are molecules expressed and secreted by microbes that enable them to colonize the host, that is, to establish themselves within the host, evade or inhibit immune responses of the host, so they're not removed, enter into or out of cells, and or obtain nutrients from the host. And virulence, again, is the degree of pathogenicity. So as an example in pathogenicity, if we look at two species of Staphylococcus, Staphylococcus aureus is the bacterium that typically causes abscesses of the skin or a wound, and an abscess is a pus-filled inflamed lesion. So Staphylococcus aureus, which typically causes abscesses, is more pathogenic than Staphylococcus epidermidis. Same genus, but Staphylococcus epidermidis is normal microbiota of the skin, and only becomes harmful, again, under special circumstances where it gets where it shouldn't be, or the body becomes immunocompromised. Now, we also mentioned back in Unit 2 that many of these virulence factors that enable bacteria to colonize the body and or harm the body are the products of quorum sensing genes. So remember, many bacteria use quorum sensing to sense their own population density. They communicate with each other by means of secreted chemicals, and that allows them to either behave as individual bacteria or as populations of bacteria. And this often plays an important role in pathogenicity and survival of many bacteria. So we know that the genomes of pathogenic bacteria, when compared to those of similar non-pathogenic species or strains, often show extra genes coding for these virulence factors. Again, the factors that enable a microorganism to colonize our body, to evade or inhibit immune responses, to enter into or out of human cells, and or to obtain nutrients from the host. And we've already learned about a number of virulence factors back in Unit 1, things like bacterial capsules, adhesins, the type 3 secretion system, invasins, and toxins. We also mentioned in Unit 2 that most of the genes coding for virulence factors in bacteria are located in special segments of DNA called pathogenicity islands, or PAIs. And these can be transferred from one bacterium to another by horizontal gene transfer. So these pathogenicity islands can be located in a bacterial chromosome or in plasmids, or maybe even in bacteriophage genomes that have entered the bacterium. And the most common way in which these pathogenicity islands are transmitted from one bacterium to another are by conjugative plasmids. The self-transmissible plasmids we learned about under horizontal gene transfer that directly transfer plasmids from one bacterium to another by cell-to-cell contact. So let's look at the terms infection and disease, which we'll be using quite a bit. Now, an infection is when a microorganism has established itself in a host. We say that it has colonized the host. So colonization is when a microorganism establishes itself in the host. It's not being removed by the host. Now, that can occur whether or not it's causing harm or imparting damage. So an infection simply is a microorganism establishing itself in the host or colonizing the host with or without causing harm or imparting damage. Whereas a disease is where there's impairment of host function as a result of damage or injury. So disease is when a microorganism is causing harm to the body. Now, the microbes that constitute the body's normal flora or microbiota have infected the body. They've established themselves in the body. But they seldom cause disease unless they invade a different part of the body where they don't normally reside, like the fecal bacteria getting into the urinary tract, and or the host becomes immunocompromised. And the term etiology refers to the cause of disease or pathologies. So in infectious disease, the etiologic agent is the microorganism causing the disease. Now we can divide diseases into a number of different categories. A communicable disease is one that spreads directly from person to person. A few examples of that. Chickenpox is caused by the varicella-zoster virus, or VZV, and this is transmitted by inhaling or contracting viruses released from the blister-like lesions seen during chickenpox. Typhoid fever is caused by Salmonella typhi. It's spread by the fecal-oral route. Tuberculosis caused by Mycobacterium tuberculosis is transmitted by respiratory drops. And of course, we could add to that COVID-19 caused by the SARS COVID virus 2. Now, contagious disease is a communicable disease, but it's spread easily from person to person. So it's highly contagious or spread easily. Examples would include influenza, caused by the various influenza viruses, again transmitted by respiratory droplets. Measles, caused by the rubeola virus, transmitted by respiratory droplets. And of course, COVID-19, caused by the SARS-CoV-2 virus. Now, a non-communicable disease is one that will not spread directly from person to person. And there's a number of examples of that. Some diseases are due to exotoxins, where it's a toxin that the bacterium produces that's causing the harm, but not an organism actually infecting the body. For example, tetanus is caused by Clostridium tetani. And this is transmitted when endospores of Clostridium tetani enter an anaerobic wound. Remember, Clostridium tetani is an obligate anaerobe. Under those conditions, the endospores germinate, And as the vegetative bacteria grow, they produce an exotoxin called tetaноspasmin. And this exotoxin, once released by the bacteria, causes a spastic paralysis, which is a contraction of the involved muscles where they contract and don't relax. So tetanus is not communicable. You have to have the endospores entering an anaerobic wound, then they have to germinate and produce the toxin. But the bacterium itself is not transmittable. Botulism is caused by Clostridium botulinum, transmitted when Clostridium botulinum endospores germinate in improperly canned foods. And once these endospores germinate, the vegetative bacteria grow in the food and produce an exotoxin called botulinal exotoxin. And when that exotoxin is ingested with the food, that leads to a flaccid paralysis, which is a weakening of the involved muscles. Lyme's disease, caused by Borrelia burgdorferi, is transmitted by infected deer ticks. So those would be examples of ones that are not directly communicable from person to person. Now, many infectious diseases are acquired while seeking medical diagnosis or treatment for unrelated diseases. And these have classically been called nosocomial infections, which refer to infections acquired while in the hospital. But of course, today we have many other places where one can seek medical treatment besides a hospital, from walk-in clinics to nursing homes or any other place where healthcare is administered. And so more frequently, these infections are being called healthcare-associated infections, or HAIs, where you get the infection as a result of seeking health care. Now, two other terms we'll be using a lot, and especially in our case study-based identification of microbes labs, are the terms sign and symptom. A sign is an objective indication of some medical fact or characteristic detected by a healthcare professional during a physical exam. So a sign is something that a healthcare professional is measuring during a physical exam. Signs would include things like blood pressure, respiration rate, pulse rate, and temperature. A symptom, on the other hand, is a condition experienced and reported by the patient, such as I have diarrhea, or I have pain, or I have a fever. Now, there's a number of things that a microbe has to do to cause an infectious disease. First of all, they have to maintain a reservoir before and after the infection. So a reservoir of an infectious agent is the habitat in which that microbe normally lives, grows, and multiplies. And those reservoirs can include humans, animals, and the environment. Many common human infectious diseases have human reservoirs and are transmitted person to person without any intermediaries. Now, some examples of human reservoirs that transmit disease would be, of course, sexually transmitted diseases, measles, and most respiratory pathogens, and strep throat. In this case, the human is the reservoir for the microorganism. Some infections are transmitted from an animal to a human. So whenever a disease is transmitted from an animal to a human, we call that a zoonosis. Examples of zoonoses would include rabies, plague, and most salmonellosis. Most salmonella infections are from ingesting food or water that's been contaminated by feces from animals containing salmonella rather than humans containing salmonella. And of course, the environment can also be a reservoir for some infectious agents. Some are fungal infections like histoplasmosis and coccidioidomycosis occur when mold spores are inhaled from the environment. Another example would be Legionnaire's disease, where often that's associated with water such as cooling water that is infected with Legionella pneumophilus, the organism that causes Legionnaire's disease. So the organism has to have a reservoir somewhere, a human or an animal or someplace in the environment where it normally grows, it normally lives, it normally multiplies. Then to cause disease, that organism has to leave the reservoir and gain access to a new host. So the way in which an organism leaves a host is called its portal of exit. That's how it leaves the host to be transmitted to a new host. So, of course, the portal of exit for most respiratory infections would typically be the mouth or nose. For gastrointestinal infections, the portal of exit would be feces. And there's a number of modes of transmission. Direct contact is through things like skin-to-skin contact, kissing, and sexual intercourse. For example, some Staphylococcus aureus infections are due to direct contact either with a healthy nasal carrier and the bacteria gets onto the skin, or from direct contact with an infected staphylococcal abscess. Infectious mononucleosis can be spread by saliva and other respiratory secretions, often called the kissing disease because it can be transmitted that way. And of course, any of the sexual transmitted disease like gonorrhea or syphilis. Another mode of transmission is direct droplet contact. This is the case of aerosols produced by sneezing and coughing. So examples where you have direct droplet contact spreading the infection would include tuberculosis, measles, colds, diphtheria, meningococcal infections, pertussis or whooping cough, and of course COVID-19. Indirect transmission of an infectious agent is from a reservoir to a host by suspended air particles, inanimate objects, or vectors. Now, in terms of airborne transmission, this can occur when infectious agents are carried by dust or droplets that are suspended in the air. Some respiratory infections can be transmitted this way, although most are transmitted actually by contact with infectious mucus, things like the common cold, but others are more commonly transmitted by droplets suspended in the air. And that can, for a short period of time, also apply to COVID-19. Inanimate objects involve things like water, food, blood, and fomites. Now, a fomite is an inanimate object, things like toys, handkerchiefs, beddings, towels, or clothing that become contaminated with microbes and transmit the infection that way. Diseases like cholera, salmonellosis, typhoid fever, listeriosis, hepatitis A, these are all transmitted by the fecal-oral route. So virtually all intestinal infections are transmitted by the fecal-oral route, where the portal of exit is feces and the portal of entry is the mouth. On the other hand, things like contaminated towels or contaminated bedding might transmit Staphylococcus aureus from skin abscesses onto the contaminated object to another person who's sharing a towel or bedding, or the human papillomaviruses that cause warts. Blood might transmit HIV, hepatitis B virus or HBV, and the hepatitis C virus or HCV. And then vectors are things like ticks, mosquitoes, and fleas that transmit the microorganism to the human. Things like Lyme's disease transmitted by the deer tick, malaria transmitted by an Anopheles mosquito. And then the manner in which the pathogen enters a susceptible host is the portal of entry. So of course, the portal of entry for most respiratory infections is the mouth or nose. For gastrointestinal infections, the portal of entry is the mouth. But that portal of entry has to do several things besides simply allowing entry of the organism into the body. That portal of entry must provide access to tissues that have the correct physical and chemical environment for growth of that organism. It has to be an environment that has the right oxygen content, the right pH, the right nutrients, the right temperatures and such that are needed for the organism to survive and multiply. And again, that's why many organisms that enter our body, such as through breathing and eating and drinking, don't cause disease, because although there's an entry for microbes, they get to the wrong physical and chemical environment. There's not the proper amount of oxygen. There's an improper pH. The right nutrients aren't available. There's an improper temperature, things like that. Now, once the organisms enter the body, they have to adhere to cells of the skin or mucosa in order to colonize the body. So almost every part of the body has a mechanism for flushing more microbes out or off of the body. For example, we constantly shed the epithelial cells from our skin and from our mucous membranes, and microorganisms attached to those cells are then shed from the body. Urination, of course, flushes out the urinary tract. Feces through defecation flushes out organisms out of the GI tract. Coughing and sneezing, vomiting and diarrhea, those are all means of mechanical removal. So unless the microorganism can replicate fast enough to replace those being flushed out, which is often the case of much of our normal microbiota that colonize the lumen or cavity of our intestines, that organism has to adhere to the epithelial cells of the skin or the epithelial cells of the mucous membranes in order to colonize and resist being flushed out by the body. And of course, as we mentioned, that body environment has to have the correct nutrients, the proper amount of oxygen or lack of oxygen, the right pH, the right temperature to support growth. And of course, keep in mind the body has excellent immune defenses. So anything a microbe can do to resist our body's innate immunity or our body's adaptive immunity is going to help promote colonization of the organism within our body. And then, of course, when the organism is not just infecting but causing disease, it's going to cause harm or damage to the body. So an infection, again, is where the microbe has established itself in the host. When the organism is inflicting damage to the host, we call it disease. So what we're going to talk about in all of Unit 3 is bacterial pathogenesis. So anything a bacterium can do to aid in the requirements needed to cause infectious diseases, like those mentioned above, will influence its ability to cause disease. And as we mentioned, bacteria often carry out these requirements as a result of their virulence factors. But we have to keep in mind that there's a number of things that determine whether or not a person actually gets an infectious disease when exposed to an infectious agent. So whether or not a person actually contracts an infectious disease after exposure to a potentially pathogenic bacterium depends on several things. It depends, of course, on the microorganism itself. What virulence factors does it have that are going to aid it in colonizing the body and then eventually causing harm? But it also is going to depend on the number of bacteria that enter the body, as well as the quality of the body's innate and adaptive immune responses. So for example, if relatively few bacteria enter the body, and the body's natural immune defenses have a much better chance of removing them if they're few in number, before they can colonize, multiply, and cause harm. On the other hand, if a large number of bacteria enter the body, they may overwhelm our body's natural defenses and they may not be as successful. And likewise, a person that has good innate and adaptive immune defenses will be much more successfully in removing potentially harmful bacteria than a person that is immunocompromised. So anyone who's immunocompromised, of course, is much more susceptible to infection. Highly immunosuppressed would include things like people taking immunosuppressive drugs to suppress transplant rejection. But those same defenses that are allowing us to accept the transplant are also the defenses we need to fight off infection. Or a person with advancing HIV infection, a person with immunosuppressive disorders. These become very susceptible to infections by microorganisms, considered not very harmful to a healthy person with normal defenses. So we're going to be looking at bacterial virulence factors in this unit that influence an organism's ability to cause infectious disease. And again, these virulence factors, as we've been saying, are molecules expressed by or secreted by microorganisms that help them to colonize the host, evade or inhibit the immune responses of the host, enter into or out of host cells, and or obtain nutrients from the host. And we're going to divide these virulence factors into two categories in Unit 3. For the first part of Unit 3, we're going to look at virulence factors that promote bacterial colonization of the host. So again, colonization is where the organisms enter the body and establish themselves. The body doesn't remove them all. And we're going to look at 6 factors that are going to influence colonization over the next couple of lectures. One is the ability to use motility and other means to contact host cells or disseminate within the host. Another is the ability to adhere to host cells and resist physical removal. We're going to look at the ability to invade host cells. Most bacteria don't invade cells, but some do, whereas of course all viruses invade cells. The ability to compete for iron and other nutrients. Keep in mind, the microbes are going to have to be able to compete with human cells and other microbiota for the same nutrients. The ability to resist innate immune defenses, things like phagocytosis and the complement pathways. The ability to evade adaptive immune responses. So often it's a combination of many of these factors that enable a bacterium or other microbe to colonize our body. And then the second part of Unit 3 is going to deal with virulence factors that damage us, the host. And we're going to divide those into 3 categories. We're going to look at the ability to produce cell wall components, or PAMPs, that bind to host cells, causing them to synthesize and secrete inflammatory cytokines and chemokines. And as we'll be seeing, the more bacteria we have, the more PAMPs they release, the greater chance that you're going to have excessive cytokine production, and the immune response is going to be harmful rather than beneficial. We mentioned that briefly in Unit 1 under a number of different structures that we looked at. We're also going to look at the ability of bacteria to produce harmful exotoxins, toxins secreted by living bacteria that become harmful to our cells. And finally, the ability of a few bacteria to induce autoimmune responses, where the immune response is directed against microorganisms, accidentally cross-react with our own cells, causing damage. So that's kind of our goal in Unit 3, look at the virulence factors that first let the bacteria simply colonize the host to establish itself, these 6 factors. And then finally, virulence factors that cause harm to us, the host, these 3 factors. And don't forget, there's a self-quiz at the end you can be doing to help quiz yourself, along with the self-quizzes that were included throughout this particular SoftChalk lesson.